All right, good morning. The title of my talk is, as in the program, it hasn't changed, Property, Privacy, Priority, a History of Genbank. I'd like to begin my talk with the following quote. One trait that humans may have acquired from the squirrels and the magpies is a love of collecting. This quote, coming from some naturalist, perhaps working in a natural history museum, is simply amusing. However, when you know that this quote is from Richard Roberts, it is outright puzzling. Why in the world would one of the leading experimental biologists in the late 20th century pay attention to the art of collecting, a practice that you usually think of being done by late 19th century naturalists, stamp collectors, or coin collectors? That's the puzzle I'm going to try to solve in my talk today, and I hope the answer to this question will be clear at the end of my talk. Genbank was born on June 30th, 1982. On that day, a representative from the NIH signed the following contract for it's a $3.2 million contract for the establishment of a nucleic acid sequence database. It was signed with BBNN and Los Alamos as joint partners. Today, I'm not going to reflect on these 25 years. Many people are much better qualified than I am to do so. But I'm going to focus on the creation of Genbank. I'm going to try to explain historically what Rich Roberts tried to explain genetically, namely how and why collecting became an essential practice in late 20th century biomedical sciences. I'm going to do two things here. Oh, first, I just have to, a disclaimer. I don't have any personal memory of the creation of Genbank. When Genbank was created, I was 10, and I forgot to pay attention. So my work here is based mainly on archival research and on interviews that I've conducted with some of you here, and I'd be delighted to meet other people that haven't had a chance to interview yet. So I'm going to do two things: situate Genbank in the broader history of the life sciences, roughly from the mid-17th century all the way to the present. Don't worry, I'll leave out some of the details. The second thing I'm going to do is try to show how and why Genbank came to embody the values of public science, open access, and free knowledge. These two questions are related, and I'll show you how they're related. When I talk about Genbank, I don't simply mean a database that is on some server somewhere. I think about all the research that is being also done with Genbank. So I think of Genbank as a particular and specific way of doing science. It's a specific way that is based on the following activities of describing, of collecting, organizing, comparing, and naming natural objects. This way of doing science has a name. It's usually what we call the natural history tradition. Now, you usually think of the natural history tradition, the study of whole organism. Here, it's not so much whole organisms that characterize the natural history tradition as the specific practice of describing, collecting, organizing, comparing, and naming objects of nature, sometimes organisms, sometimes bones, sometimes tissues, cells, molecules, or sometimes sequences. What I'm going to try to show today is that some of the early tensions that arose in the creation of Genbank were about questions of property, of privacy, and priority. And these tensions reflect actually the differences between the experimental and the natural history tradition. I think these different tensions prefigure current debates about open access publishing, intellectual property in the life sciences, and some of the most recent debates about open access publishing, as we've talked earlier. My talk, I'd like to say, is not about distributing individual credit. Many important actors in this story will be left out for the sake of clarity, but it's really about trying to capture the historical significance of Genbank. So now the big picture. Experimentation is perhaps the most distinctive feature of modern science. Our very idea of modern science that we trace back to the scientific revolution, to Robert Boyle's air pump on the left, to Harvey's experimentation on circulation of blood, gives a central place to this particular way of producing knowledge about the natural world. And today, the authority of science rests largely on the possibility of doing science in the laboratory. However, as we all know, experimentation has only been one way in which knowledge has been produced about the natural world in the last 3 centuries. An independent tradition, natural history, has also been along. A tradition that we associate with a very different place, was the place here, like the Cabinet of Wonders here in Naples in the mid-17th century, and many of the places like gardens, museums, and other places which are centrally collections. Collection, much more than the field, is a central feature of the natural history tradition. Collections were not just repositories or places to collect, and sometimes they were, like in this example on the right, but slowly collections became tools to produce knowledge. They became tools to produce knowledge about the order of nature and how that order came about. Think of the origin of species, and also about the identity of specimen. It was by bringing a specific specimen into a collection that you could understand and figure out its own biological identity, where it did fit in the larger order of nature. The reason why many of the collections were being developed and were created was in large part a reaction to what you can call the information overload. In the 17th century, for example, the expansion of European travel brought an increasing number of new specimens, plants and animals, back to Europe, which just did not fit in the current understanding of nature. As a reaction to this increasing amount of information which was available, new collections were created to try to make sense of this great diversity of nature. Historians of science tell us that natural history and experimentation have an interesting history in the way in which they have interacted. They have lived side to side pretty peacefully all the way to the 19th century. In the 19th century, experimentation started to take over, especially in experimental physiology. It started to dominate the life sciences. In the early 20th century, think of genetics, for example, and throughout the 20th century, it became really the dominant way of producing knowledge about the world. Natural history declined and became relegated in some obscure museums and became essentially, the museum collections became essentially tools for display rather than producing knowledge, and places where you would bring your kids on a rainy Sunday afternoon. Now, the way in which early molecular biologists have told their own story matches very much the story I've told you from historians of science. Early molecular biologists, here the first time the word was used in 1938, and progressively in the 40s and 1950s, liked to show and display their contempt for traditional biology, by which they meant natural history. They were very proud, and E.O. Wilson, for example, has wonderfully described the tensions, to say the least, between molecular biologists on one side and naturalists at Harvard in the 1960s. Some of the early molecular biologists, some of which were physicists, converted to biology, some were biophysicists, were proud to quote Rutherford with his famous quote, "There is physics and the rest is stamp collecting," showing at the same time their contempt for anything else than physics and also for this traditional way of doing biology, of stamp collecting qua natural history. What is interesting is that if we look at today at the forefront of biomedical research, biological collections play an essential role. Genbank is just one example, perhaps one of the best known examples. Collections and database are really at the forefront of research today. So just this simple fact forces us to rethink the big historical picture that I've just outlined earlier. There is a puzzle to be solved of how and why natural history, which declined throughout the 20th century, which was the thing that molecular biologists really did not want to do, these practices came to be so central for our understanding of nature. There seems to be today a new alliance between natural history and the experimental tradition. A new alliance, however, that has not always been smooth and that had to be built, for example, around the creation of Genbank. This new alliance also showed and displayed some tensions over different ideas about what knowledge, how knowledge is produced, and what it is for. And these tensions, I think, illuminate some of the early history of Genbank. Now, there's no single starting point to the history of Genbank, or there are perhaps many. I'd just like to start, like Rich Roberts did, with a meeting that took place at the Rockefeller University in March 1979. This meeting, the agenda of this meeting, which was set up by Norton Zinner and Carl Anderson from Brookhaven, the goal of the meeting was to discuss ways to collect, verify, and make available to the worldwide scientific community nucleic acid sequence information. There are about 30 participants at the meeting with very different backgrounds. On one side, molecular biologists like Rich Roberts, Alvin Kabat, Fred Blattner, people who are interested in computer applications. Josh Lederberg, he was of course also a molecular biologist, but the last 20 years he had really been pushing computers in the life sciences. Cy Leventhal and the younger generation, people like Temple Smith, and a third group of people who were interested and who were actually maintaining biological database. Margaret Dayhoff and Olga Kennard from Cambridge. Representatives from the NSF and from NIH and the Department of Research Resources were also present. At the end of the day, the meeting concluded that a centralized data bank is essential for the efficient use of nucleic acid sequence information. There seemed to be a consensus on that point. Again, what had prompted the meeting and what had prompted this strong consensus was, as in the previous natural history tradition, information overload and the heuristic value of a centralized collection, so what you could do with a collection. As you all know, and you just remember here, that the new DNA sequencing methods were just being applied just after 1975 and 1977, when Fred Sanger and Al Maxim and Wally Gilbert published two new methods of sequencing. And that's the result in the number of papers that are published and that contain sequence. So you can see here in 1978, you see the curve is starting to grow exponentially. In 1979, when the meeting is taking place, there might be already a feeling that the number of sequences is going to become extremely large in the near future. But also, what was probably most interesting was everything you could possibly do now if you had such a sequence, a database. Of course, the questions of molecular evolution, which had just started to become important in the mid-1960s, were becoming increasingly so in the 1970s. The mechanism of molecular evolution was a heavily debated topic in the 1970s, especially after the neutralist theory had been presented. Analyzing gene structure became also a fascinating topic after 1977, so just 2 years prior to the Rockefeller meeting. The intron-exon structure was figured out, Tata boxes, and other fine structures of the gene were being published. And also, new sequencing technologies was also showing how important it would be in the near future to be able to have a database that could help you organize and analyze data. Here, this quote, I think, is revealing the rate-limiting step in the process of nucleic acid sequencing is now shifting from data acquisition towards the organization and analysis of that data. And finally, what you all think is the most important function of Genbank today, inferred gene function through homology searches, was actually perhaps the least important aspect at the time. So there was a strong consensus in favor of a national, computerized, and non-proprietary database. However, there were also strong disagreements at that meeting. Disagreements and tensions also around ideas that a centralized database would constitute a threat to individual collections, the existing collections at the time, which would be rendered obsolete. Other participants were concerned about how to protect privacy and privacy of data. Did it have absolutely to be made public right away? Would that establish priority if you would submit data to a database? And finally, fair access to the collection was also a major concern. Would the institution or laboratory hosting the national database have some kind of privileged access to the data that had been produced by the community? So all in all, these different tensions and these concerns, I think reflect the idea that one of the key issues was how to balance individual and collective interests. In the 1980s, a number of workshops and meetings were held at the NIH and at the EMBL. I'm not going to go over them, but to try to define the scope of the database, organization, how to collect, and how to distribute data. Ruth Kirstein has already been mentioned, was resistant at the beginning, and then took on the project, and Elke Jordan played an extremely important role in setting up the framework at the NIH to fund, eventually, Genbank. What is important, I'm going to leave out the EMBL part of the story, but in June 1980, the EMBL announced that it would make its own database available once it got so far at having it together in an acceptable format. That triggered an immediate response here at the NIH. Elke Jordan wrote the same month, "Clearly, we must act now." A sense of competition seemed to grow between the European group and the American group about getting first, at some point, a public database out there. In December 1981, to make a very long story short, the NIH issued this request for proposals, a complex funding scheme by NIH, different institutes, the NSF, the NCI, Department of Defense, and the Department of Energy. So all this had to be figured out. That explains also part of the delay, if you want, between the Rockefeller meeting and 1981. People in the scientific community at the time complained about the NIH being slow in taking the lead, and also pointed to the bureaucratic inertia of the National Institutes of Health. It's not my role here to judge if the NIH was slow or not. I'm not sure what the benchmark should be, but there's one historical important point here that's often overlooked. It is not at all clear, it wasn't clear at the time, that a collection was something of importance, of any scientific importance. It wasn't clear that that would represent 10 or 25 years down the road, the forefront of biomedical research. Here, I think Fred Sanger put it in a nutshell: doing for a scientist implies doing experiments. So it wasn't clear that just collecting stuff was actually qualified as good science and therefore should be funded by the NIH. Several proposals came into the NIH, and two of them made the cut to the second round, one by Margaret Dayhoff from the National Biomedical Research Foundation, and one by Walter Goad from Los Alamos National Lab and BBNN with Howard Bilofsky. They were teaming together. I'm briefly now going to present these two players and try to show in what stage they were in their own development when they submitted this proposal to NIH. Margaret Dayhoff got a PhD in physical chemistry in 1949 from Columbia University. She used punch cards, so for those of you who remember these, punch cards to calculate, punch card machines to calculate resonance energies in small molecules. She joined in 1960, she joined the National Biomedical Research Foundation, a private non-profit research organization which had just been created. It had been created by someone called Robert Ledley, who had a degree in, who was a dentist first and foremost, and eventually got a degree in physics, and became one of the proponents of using computers for all kinds of things at the beginning. So he was really interested in computers and was driven by the desire to develop computer applications and early digital computers. He was made a member of the RNA Tie Club in 1954. It's an important club which never met, met in part, which was organized by George Gamble to try to solve the genetic code theoretically using pencil and paper. You can recognize Francis Crick, Jim Watson, Alex Rich, and Leslie Orgel on this picture. The reason why he was hired in that group is because of his expertise in the field of computers, something that not everybody had at the time. So that's how he got interested in biology. He got interested in sequences, and eventually leading his interest into applying computers to biology and medicine. He became the author, and he was perhaps in the 20th century one of the key persons that pushed computers in biology and medicine. He was the author of this 900-page monograph, which surveyed all the possible uses of computers in biology and medicine, which was published in 1965. So he created also, to further this goal of pushing computers in biology and medicine, that's why he created the National Biomedical Research Foundation. He hired Margaret Dayhoff, and the first project she worked on was to develop a computer algorithm to assemble partial protein sequences, of course, at the time. In order to test her algorithm, the first thing she did was to try to collect the available sequences. So she tried to collect by scanning simply literature and find all the sequences available. She got these sequences together eventually in a book called The Atlas of Protein Sequence and Structure, published for the first time in 1965. And this is how she writes to a colleague and explains why she thinks it's important to collect sequences. There's a tremendous amount of information regarding evolutionary history and biochemical function implicit in each sequence, and the number of known sequences is growing explosively. We feel it is important to collect this significant information, correlate it into a unified whole, and interpret it. So this was the goal that she set for herself by publishing this atlas and is in several subsequent editions. A typical page would look like that. You would have simply a sequence with a 3-letter annotation, and then you would have the 1-letter annotation, amino acid composition, and one reference to where she got the data from. She also developed different sequence analysis methods, especially for evolutionary studies, the PAM matrix being perhaps her best known contribution. The way in which she got the data was by surveying manually the literature, flipping through pages of journals at a time when the word sequence was usually not even indexed. The number of known sequences was growing explosively at the time, putting an increasingly heavy burden on the search for all the sequences. To give you one example, the 1965 edition of the Atlas had only 100 references, and in 1972, it was one order of magnitudes higher, 1,000 references. She was hoping to base the collecting system on a gift economy. By that, I mean that she encouraged researchers to send in the data before they were published, and in exchange, they would get a free copy of the Atlas. Needless to say, the system did not work. Some people did send in sequence, but most of them did not. At the same time, the NIH was kind of hesitant in funding Margaret Dayhoff's collecting efforts. The NIH funded very generously the phylogenies that she was working on, but was not really sure whether it was the role of the NIH to fund the collecting enterprise. this, I think, points again to the doubtful value of collections and collecting as a meaningful scientific activity in the 1960s and 70s. I think there's an important letter from John Edsel that he writes to Margaret when she believes she would like to become a member of the American Society of Biological Chemists. And Edsel replies, I think you're the right kind of person. However, the candidate must demonstrate that he or she has done research that is clearly his own. The compilation of the atlas scarcely fits into this pattern. So you can see how doing this kind of work doesn't really qualify as fundamental science. As a result of these funding difficulties, as a result of the difficulties of getting data in, she decided to copyright the atlas and to sell the content of her database, first selling the atlas and then eventually the magnetic tapes for a relatively small amount, but a significant amount that made all the difference between a kind of gift and free knowledge and a kind of somewhat a commercial enterprise, even though it was nonprofit. This provoked a certain amount of resentment in the scientific community, which would probably have important consequences later on. Here is someone writing to Margaret Dayhoff when he learns that he has to pay for the atlas. You're somewhat in the position of a folk song collector who copyrights his published material. Do I have to pay him if I sing John Henry? So you can see here this unease was the fact that she was actually charging for data and for sequences that she had actually got for free. In September 1980, she makes public the first nucleic acid sequence database, and she makes it public, available. It's the largest DNA database available at the time. And it's available through a modem connection. As you can see here, you put up your phone and you hook it up to your computer. It was free, but it was copyrighted. And you had to agree not to redistribute the data elsewhere. This is what you would see on your screen when you would connect on her pilot database. You're encouraged to use the retrieval system, to comment on it, and to contribute to it. You're licensed to use this data for your own research. As a licensee, you are legally obliged not to redistribute the data or otherwise make it available to any other party. The rapid expansion of nucleic acid sequences at the time and low NIH funding was a serious concern for Dayhoff at the time. Here she writes to a colleague, databases do not inspire excitement. As a result, she decides here to get the database on a business-like basis and make it available on a subscription basis. You would have to pay $1,000 if you're an individual working in a lab. If you're a private company, you had to pay anything between $5,000 and $10,000 to get the magnetic tapes. Now that's the situation when she submits a proposal to NIH. Now let's look at Walter Goad. Walter Goad was a physicist, spent most of his career at Los Alamos National Laboratory, had been involved in development of the H-bomb during the 1950s and 60s, something that disappeared from his resume later on. He was a member of the theoretical and biophysics group, the T10, under George Bell from the very beginning. It was a place where some people were interested in computer analysis of molecular sequences. People who were a member of the group and people who were kind of associated with the group more or less loosely would go there for the summer. The reason why this research took place also in Los Alamos was in part because of the unique computer facilities that Los Alamos could offer. And this would be a cornerstone of the argumentation of why Genbank should be at Los Alamos. In a letter, Walter Goad, it's not Puck, it's Walter Goad who's running this. He says, Los Alamos is the natural place to locate a center for sequence analysis of DNA, primarily because of the National Lab's unique computer facility. That was the main rationale for having it at Los Alamos. Goad started to collect, after the Rockefeller meeting, started to collect all the known sequences from other collectors, from small collections in Europe and in the United States. So in 1980, he was able to send out a memo calling for cake and coffee to celebrate the 100,000 bases now in the DNA sequence library. Dayhoff was the only person who refused to share her collection. She had the largest collection at the time. This is how a European colleague reacted when he learned about Margaret Dayhoff's unwillingness to share her collection. An important strength of your program, this is Walter Goad, is its openness. Sequences in the system will be quickly made available to everyone and will not be the private hunting grounds of the center's staff. I feel that that's in refreshing contrast to my perception of the way in which the protein sequence library is maintained by Dayhoff and her staff. You probably now have a better understanding that he did at the time of why actually Margaret Dayhoff did not actually share her collection. Now that's a situation when the proposals come in at the NIH from Walter Goad on one side and Margaret Dayhoff on the other. Now I'm going to briefly compare and contrast these two proposals and try to show you how they differed in some very significant ways. I'm going to look at collecting data, distributing data, and the question of copyright. In the natural history tradition, many strategies have been applied to try to collect data, specimens, plants of all kinds, or eventually data. The strategies included patronage in early modern Europe, for example, gift exchange between botanical gardens, expeditions to far remote countries to bring back specimens, commerce, simply buying specimens on the market. But the most important collecting strategy in the natural history tradition has been networks of anonymous amateurs. They would give freely and so often anonymously their specimens to a collector which they respected. Dayhoff pretty much thought to do the same thing. She thought that she could collect, and her group could collect DNA sequences doing a manual survey, as she did previously, but also she was hoping once again to base the collecting system on voluntary contributions, pretty much basing her collecting efforts on a communal altruism, which would move people to send in data. Now, it is clear that data did not, at the time, sending in sequence did not establish priority, and she had made this crystal clear in the very first edition of the Atlas, in the preface of which she wrote, "It is not our intention to become involved in questions of history or priority." In many ways, the system that Dayhoff imagined conflicted with the reward system in the experimental life sciences, in which you get individual credit, not collective rewards. You get credit by publishing a single paper in your own name in a journal like nucleic acid research. Unpublished sequences were considered highly proprietary at the time. People had put lots of effort, time, and money into producing the sequences, and it was only fair that they would get some reward by publishing it in a peer-reviewed journal. Walter Goad had another idea of how to collect. He thought about doing it the same way as Margaret Dayhoff, but in addition, he outlined a proposal for a mandatory submission model enforced by journal editors, pretty much the system we have today. So he was very much thinking of individual rewards for the people who would send in sequences. Here is how he writes about it in the proposal. Once a national standard is established, most journals will be willing to furnish or require authors to furnish a computer-readable copy of each sequence. He is not the one who invented this system at all. It was a system which was, I believe, first proposed at the EMBL, but which then Walter Goad made his own and tried to promote in his own proposal. He was also hoping that the rise of computer-to-computer transmission would make the whole process easier. The way in which Walter Goad thought about this whole project was very much aligned with the reward system of the experimental sciences, in which publication, I just said, establishes authorship, priority, and credit. And this was his model at the time. I think Louis Wolpert made a nice point to summarize the issue. He was talking about something completely different, but summarize this issue. In order to promote the success of their ideas and hence of themselves, scientists must adopt a strategy of both competition and collaboration, of altruism and selfishness. Each must balance his or her behavior in relation, for example, to sharing information. So Walter Goad seemed to have a pretty good understanding of how this would work and what incentives were needed to bring sequences into a national collection. NIH reviewers were very much in favor of the model proposed by Walter Goad, which they thought might prove be efficient. It's another question why the model was not actually implemented until almost 10 years later. Collection doesn't only rest on a sophisticated mechanism of reward, it also rests on trust and the authority of the collector. Most natural history collections have worked very well to collect data because they were headed by a respected naturalist. In this case, Walter Goad and Margaret Dayhoff were clearly outsiders to the biomedical community. In addition, the places where they worked was somewhat of a liability to their own project. The National Biomedical Research Foundation was a private institution, and it raised the suspicion that it might try to make profits out of a national database. Things were even worse for Walter Goad. Walter Goad was at Los Alamos, an institution that was believed to be military in some way, and everybody knew that it was a national lab. A national lab which had its own culture of secrecy and security, which would actually discourage perhaps this image of open science. Of course, they couldn't imagine that the NIH would look like that 25 years later. And here is, I think, Walter Goad was very much aware of how much this issue of secrecy and security was played out in this story. He writes here to somebody else at Los Alamos, "I have some misgivings about being in the secured area during the first 6 months of 1982. We expect to be evaluated during that time for the computer-based DNA sequence resource. It is important that we be perceived by the molecular biology community, and particularly by our reviewers, as offering completely free and open access to the information and program we'll be collecting." So it's really a question of image, and he is very aware of how important that is. Distribution of data was the second most important aspect of the whole proposal. Margaret Dayhoff proposed to distribute it on paper, which was a requirement at the time, and also on magnetic tapes. Walter Goad did the same, but he also insisted on computer networks, ARPANET and Telenet, shared computer resources as Profit and SUMX, and mini computers. So he thought that that would be an important way in which data could be distributed. This is a drawing, perhaps by Walter Goad, of another proposal that he made. It's not very important here, but here's, I think it shows the role of networks. You see Telenet here, which would help users on this side with a computer to connect to Telenet and submit data here to BBNN or IntelliGenetics. They would then pass it on here to Los Alamos using ARPANET on this side. Curators and people who were doing annotation would also use Telenet to access the Los Alamos computers. So he was very much aware of the importance and the growing importance of networks at the time. Walter Goad outlined his willingness to promote electronic communication and collaboration among users and offer online access to the Los Alamos facility over national networks. This is just a map of what ARPANET looked like in 1980, so connecting some of the main research centers in the United States, as long as they had some department DoD project. The reason why this is important is not just because it provided a handy technical mean or way to share data, but it also highlighted the importance of having completely open access to the data, which was the very idea on which scientific objectivity rests, which is this unlimited access to data. The cultural values of networks and user groups, such as GenNet at the time, which had just been closed down partially, and BitNet, was also on the rise in the early 1980s. The third and perhaps most important issue for the NIH was the question of ownership of data. Who would own the data? Natural history collections have almost always been proprietary. They belong to some collector. In the experimental tradition, on the other hand, published knowledge was often thought to be public, something that there shouldn't be any barriers to the circulation of knowledge. So the greatest NIH concern at the time was who would copyright and who would own the copyright and the ownership of the data in the future database. The review committee put this most clearly: will NIH own all the data in the database? Walter Goad was unambiguous in his reply. He said he did not intend to assert any proprietary interest whatsoever in any data. And in case the reviewers missed the point, he contrasted his model with the one of the National Biomedical Research Foundation. He said the Los Alamos database was freely available to anyone without restriction on further distribution, unlike the NBRF team, who has fought revenues from sales of the database and prevented redistribution. So he drew a sharp contrast between both. Margaret Dayhoff was more ambiguous about whether or not she would still hold a copyright, or the NBRF would hold copyright on the data which was collected prior to an eventual NIH contract. The reviewing committee found that Margaret Dayhoff's answer was not very reassuring, and saying that altogether the NBRF's previous proprietary arrangements are not reassuring. Walter Goad was again aware of the edge he was seeming to develop. He said he was seeming to be developing an edge as our principal competitor is becoming increasingly enmeshed in proprietary arrangements. In June 30th, 1982, the NIH awarded a $3.2 million contract to Walter Goad, BBNN, and Los Alamos. Now, I'd like to conclude here by a couple of remarks on what we can learn from this story. I think the first important thing is that the creation of Genbank was predicated upon the resolution of tensions between these two traditions, the experimental tradition and the natural history tradition. These tensions were over issues of privacy, property, and priority that still play out today. Margaret Dayhoff aligned very much her collecting enterprise on the values of the natural history tradition. Walter Goad, on the other hand, was very much more aware of the values which guided the experimental science tradition, which made much more sense since that was where the data was actually coming from. Genbank came to embody specific values, the values of public science, of open access, and free knowledge. It was in this very specific context and in these tensions between Walter Goad and Margaret Dayhoff that these values came to be key principles for Genbank. That was made possible by some key individuals, but also by a very specific historical context of the early 1980s, before gene patenting became a big issue and before private genome sequencing projects were around. So it was in that particular window of opportunity that Genbank came to embody these particular values. Genbank today represents probably a new articulation of this natural, historical, and the experimental tradition, a new articulation that is just starting, I believe, to transform the way we do science and our understanding of life. I thank you for your attention.